merci Sirlifi pour l'introduction. Je remercie énormément les organisateurs pour m'avoir donné l'opportunité de venir visiter Constantine tout juste après la neige. C'est vraiment très joli à voir et très intéressant. En venant du Mexique où il fait 25 degrés, ça m'a un peu rafraîchi les idées. Si Ibn Qasim, merci de m'avoir suggéré. C'est vraiment un grand plaisir de venir voir. Et finalement, hier, j'ai eu l'honneur de voir le Khroub. C'est une station très importante pour le CIMMYT. Donc, aujourd'hui, je vais parler un peu de l'aspect pratique de l'utilisation de la séquence de la séquence de blé qui vient d'être publiée. Donc, je ne vais pas parler du côté technique, du côté séquence lui-même, mais surtout de ce qu'on peut faire avec cette nouvelle avance technologique. Je vais continuer en anglais si ça ne vous dérange pas. Okay. All right. Thank you, everyone. I'll be talking not really about the sequence itself or the technological advance that constituted this publication of the sequence, but rather on the utilization in breeding, including at CIMMYT and other places, of this new tool that has been made available to us. All right. First of all, I would like to acknowledge and thank my colleague, Dr. Susanna Dweisigacker. She's the head of the molecular marker lab for wheat at CIMMYT. She's the one who is in charge of doing most of the genomics work in our institution, and she certainly has been a very, very interesting person to work with and a very good collaborator for the breeding program. Her mandate is basically to do anything about marker, either in terms of research, developing markers, but also to apply markers that have been developed by others in our breeding program, and then guide us and help us in the breeding program. So it goes all the way from services, all the way to research and marker development and training as well. Okay, so the reason we're here is to kind of understand what are the implications of this publication here, of the complete order and annotated sequence of bread wheat that was published in 2018. And this is definitely an important step for all of us. It's an important step, first of all, because it's a big and very difficult achievement. Wheat, as was said previously, is not a small genome. It's a huge genome. One chromosome, just the 3B chromosome of wheat, is bigger than the rice, the entire rice genome. And it's a very complicated genome because of this polyploidy, the presence of a lot of translocation, mutation in different areas, a lot of repetitive DNA, and a very, very high number of genes. So it's really the Mount Everest of genomes, and we finally finished climbing it last year. Just a few follow-up measures that have been taken after the sequencing of the China Spring variety is what they call the 10 pan-genome project, where they're sequencing different variety to finally go beyond one reference sequence, but try to look at different variability, allelic variability in different gene pools. And so different countries have contributed different variety that are now being sequenced, and more are coming. So with the advance of the technology, more people are submitting variety to be sequenced. And so we're going to have really a wealth, a very, very important amount of data that we can be using very soon. But I'm a Durham breeder, and to me, really, the most important thing is Durham wheat. I was interested by the bread wheat sequence, but I was really, really thrilled by the prospect of publication of the Durham wheat genome, the cultivar Svevo. It's an Italian variety that has half CIMMYT blood. And this thing has not been published yet. It's still in press, but it's just been accepted for publication in Nature Genetics. And really, it's a consortium of very different people from different countries that have finally finished the sequence, but also did some analysis that's really interesting in terms of the domestication of Durham wheat. So this is really very exciting for me, and we have already used it. This sequence was available last year, just that it took a little bit of time for the publication to come out. Now, as breeders, why do we use the sequence? Why is it even interesting? It's just how are we going to use it? How is it going to change our lives? and our way we work. After all, traditionally, breeders have had very, very big success just using phenotypic selection. I mean, we've raised yield potential by many-fold using only phenotypic selection. We have introduced very different disease resistance. We have improved quality very significantly over the years using only phenotypic selection. In fact, there was even some really big revolution that happened without the knowledge of the sequence. In fact, when people have transferred, for example, SR2, a gene for stem rust resistance, from a wild species into wheat, people did not even know what the structure of DNA was at that time. The green revolution that has completely changed wheat worldwide has been done at a time where molecular markers, DNA was not even known really how it was structured. So there was a lot of really big progress that was made without the knowledge of the sequence. So what has changed now? In order to understand that, we have to really look and think about what are the limitations of phenotypic selection, okay? And there are many circumstances or instances where phenotypic selection alone has been limiting for progress. Think of, for example, if you want to pyramid 2 genes of resistance to rust, to septoria, to any diseases. If one gene gives you 100% resistance, that you will never know if you have another gene that is accumulated or a third gene and so on. So for those situations, phenotypic selection is not very effective. And sometimes, not sometimes, always, when you rely on phenotypic selection, you have to have a selection pressure. You have to have a pathogen, you have to have a drought and so on, and you don't always have that. So sometimes you lose opportunity for selection because the selection pressure is not there. Or sometimes you are threatened by a pathogen, for example, that is not in your country or in your region, like in the case of the UG99 races of stem rust. They are in Africa, they're not in Europe, they're not in the Mediterranean yet. But we need to be able to breed for resistance to those pathogens, because eventually they will come, and there is no way to do it phenotypically if we don't have the pathogen here. You can go to Kenya and do your breeding program, but that's a little bit of a problem logistically. And then you have traits that are important, but that require a lot of resources to be phenotyped, or that require for you to go to uniformity 5-6 years of breeding before you can actually phenotype these research and rely on phenotypic selection. So all of these situations are limiting in terms of the capacity of phenotypic selection to really result in a significant progress. So really, what is important about knowing the sequence of wheat, the whole sequence, is that it allows us to it gives us access to the possibility of producing molecular markers. Now, what is a molecular marker? For those of you who really don't know about this, it's basically just a DNA sequence of known location in the genome that is polymorphic between individuals, and that can be visualized by any lab technique that is available. And it can or cannot be linked to a gene of interest. In terms of breeders, we like those markers that are linked to gene of interest. For other people, it's different. So yeah, these molecular markers have been present for, 30, 40 years now, different types. People have put markers on genetic map, or have used markers to develop genetic linkage map, first of all. There were very few markers in a very big genome. But then they went to physical mapping partial parts of the genome, and that's a more precise location where you do not rely on the recombination between markers to look at the distance, but you actually count the number of base pair, and it's actually physical distance. So that's an extra level of precision that started a couple of decades ago. But really what the complete sequence of the genome affords us or gives us is the entire sequence with all the structures of the genes that is present in an organism. And that gives us a lot more precision, and then opened up a lot more possibility to develop molecular markers that are useful in our breeding programs. Now, all markers
even 20 years ago, even starting from RFLP or SSRs or STS, require some knowledge of sequence. But only partial knowledge was enough at that time. Now we have the knowledge of the entire organism. So basically it gives us unlimited capacity to develop molecular markers. All we have to do is, okay, where is our gene? And now we can find markers to be able to monitor this gene when we select for desegregated populations. So this is the most important thing. And we do that by looking at these partial sequences that we know of and aligning them into the complete sequence, and then it tells you where we are exactly on the genome. And we can look in the neighborhood of the genes that we have, where to find good markers, and even we can come up with ways to come close to having perfect marker right in the genes if we know the kind of the structure of the gene that we're looking for. So unlimited source of DNA markers and faster identification of candidate genes. So if you're looking for disease-resistant genes, we know through genetic studies that disease-resistant genes have a certain type of structure. Gene families are known. And so by looking in an area of the genome that co-segregate with the disease resistance, we can see maybe limit the number of genes that could be involved to a dozen or 2 dozen as opposed to hundreds before. Okay, so all of this is important. Why? Because in many breeding programs, especially big breeding programs that have global interest, like the CIMMYT program, like the ICARTA program, like the program of the multinationals, people who work in different areas of the globe, we have started to rely on molecular markers very heavily in our breeding program. These are the number of molecular data points generated just for marker-assisted selection at CIMMYT. The total is last year more than 200,000 data points that we have generated. That's roughly about 90,000 plants that we looked at with gene-based molecular marker. We're not talking about entire genomes, but with gene-based molecular marker to aid us in our selection. So we select the plant that actually have the genes that we want or have the combinations of genes that we want. Okay? There is many genes that, many markers that we use for many genes. Rust resistant is probably the most important one. So we can pile up as many resistant genes as we want. So our resistance lasts more and is not overcome after 2 to 3 years, but stays longer. There's many sources of genes that have been marked now and is routinely used. This is rust resistant in bread wheat, for example. Fusarium head blight is another one where we know some of the loci that confer resistance to Fusarium head blight in bread wheat. We have been able to develop, thanks to our collaboration with our people in China, to come up with markers that are even closer to the genes and are more reliable for marker-assisted selection for this disease. It's not just disease, but it's also about quality, nutrition quality, zinc concentration, which is a very important trait, is polygenically controlled, and we have been able to identify some QTL as some genes, and now we have the marker that will allow us to actually increase the concentration of zinc in our selection. One that is very important to me, that's seed size. Either monitor a mutation through molecular marker or looking at QTL, marker that links to QTL for increased seed size, and that's important because you can do selection for seed size early on, and that is a mechanism either for higher yield but also for drought tolerance, and this is something that we've been using very effectively and very extensively in the last 2 or 3 years at CIMMYT. In my program, in the Durham breeding program, also we have come to rely a lot on molecular markers. Last year alone, we've isolated DNA from more than 21,000 plants just to be able to do marker-assisted selection, generating some close to 50,000 data points. So it is about 20% of the crosses we make or the populations we handle are handled through phenotypic selection, complemented by marker-assisted selection. These are the list of the traits or of the genes we follow or monitor. We select based on molecular markers. If you're interested in knowing more about that, I can talk to you about it later. This is just to show you that it goes from rust resistance to quality to grain size. Again, some numbers. If you look at the majority of our work is for rust resistant, pyramiding different genes of rust resistance. That includes stem rust. And in the case of stem rust, we're pyramiding genes for a disease that is not present in Mexico, so we're doing preventive breeding here to be able to be relevant to Ethiopia, to parts of India where races are present. We do a lot of marker-assisted selection for quality, diversifying the gluten protein of durum wheat, which have very little variability compared to bread wheat. We're using marker-assisted backcrossing to introduce bands from bread wheat or from different land races. We're making durum wheat soft. We're introducing the thin gene or the softness gene into durum just to explore how useful it could be. And so these are a little bit some of the traits that we follow in using marker in the durum program. Now, just to give you an example of how useful knowing the full sequence of durum wheat was for us, and how before getting the sequence we could not do these kinds of things easily. Now we could do in a couple of months what we couldn't do last year before the sequence was available. There is a gene, a leaf rust resistant gene called LR14A that's very important in durum wheat. It's a gene that has broken down in France, it has broken down in North Africa a couple of years ago, but it's still effective everywhere else. And it's very dominant in our breeding program, whether it's the CIMMYT gene pool, the Icarda gene pool, the European, North African gene pool. It's just a gene that is present there. We want to get rid of it because we know it's going to be ineffective. So in order to get rid of it, we need to have a good marker for it. There is an SSR marker that is more semi-diagnostic, let's just say, but we wanted to come closer to the gene and get a much better marker that could be used in a high throughput platform. So we took two sister lines, one with the marker, and both of them with the marker, one that's resistant, one is susceptible. We developed some population, Rails population, not Nails population here, if that's a mistake. And then we divided this population based on phenotype, the resistant group, the susceptible group. We bulked the DNA from each group and then looked at GBS marker very fast, very cheap, with a collaborator in Kansas. And then once we had that, we looked at the GBS sequences or tags, these markers that are very good because they're very cheap and very fastly done. But once you've got these markers, it's very difficult to use in breeding for marker-assisted selection. You have to switch to a different platform that is more easily used and that provide you markers that you can actually use in marker-assisted selection. But these GBS or genotype by sequencing markers or tags can be aligned into the wheat genome. And I'm sorry, it's a little bit complicated, but this is an output of the software. The lines over there on the top are basically, think of them as ATGCs, just the big sequences. They're sorted 100 by 100. And so we have the entire sequence there. We have the GBS tag, the left and the right one, and the sequence of our bulks, of the one that is segregating in our population. So we aligned it to the reference sequence, and then that allows us to look at different markers, SNP markers, that can be really rapidly automated. But not only that, we can also look at the genes in the reference sequence, and then identify genes that are likely, because of their structure, to be disease-resistant genes that pertain to families that are known to be resistant genes. So, at the end of the day, after a couple of months of work, and not a lot of work in-house, mostly outsources, Susanna came up with 19 cask markers to validate, and she's currently validating them on the entire population. And out of those 19, maybe 2 or 3 will be really very close to the gene, and we could sell it to a private company or do it in-house, and we can run thousands and thousands of samples of the markers, and then really come with results very fast and a very cheap way. This thing would have taken months, if not years, if we didn't have the reference sequence. So basically, things that used to take years to do and that was a little bit difficult to do can be done in a couple of months, thanks to this roadmap that we have now. So it makes us a lot more efficient, a lot more effective, and a lot faster in the developing of markers. Okay. Anyway, in conclusion, marker-assisted selection is here to stay, I think. It is a very important and very effective complement to phenotypic selection. And in some cases, it does not even replace phenotypic selection for traits that are very difficult to select for visually. It allows a very targeted gene-based selection. And it's getting cheaper and cheaper. So access to markers is getting cheaper and cheaper. And in fact, with all the companies, the private companies that are actually provide this as a service, national programs or breeding programs do not even have to develop a lab infrastructure, which could cost millions. They could rely on service provider to do it. So, and with the availability of genome sequences, now marker selection can be done a lot faster because we can develop as many markers as we want relatively fast. Of course, it takes means, it takes money, it takes time, but it's a lot faster and a lot cheaper than it used to be before we had these reference sequences. Now, I've talked a lot about the positive things and the advantages of marker-assisted selection and using sequences to be able to do more gene-based selection in our breeding program, but there are also some cautions that needs to be kept in mind. Really, these technologies are useful when you have a viable, very competitive breeding program to start with. Because at the end of the day, I know I said they're cheaper and cheaper, but they're still expensive. If you invest in marker development or marker-assisted selection, and you don't have a program that actually does good for phenotypic selection, you're basically just wasting your money. It has to come on top of a program that is already viable, that is already competitive. It has to come to help, not to replace what you have. It doesn't mean it doesn't help you to select plant with 2 or 3 genes or 4 genes of leaf rust resistance if you don't have the capacity to test for yield later on. Okay? So it really needs to come up to complement a program that is already viable and competitive. You also need to have a strategy. Marker-assisted selection is not just sending a sample to a lab and then waiting for the result. You need to think, you need to have a strategy, to have a work plan from the beginning, from before you make your cross all the way to the end, how you're going to use marker, when you're going to use marker, and at every single step question whether you should or should not use marker. And never use markers if you can do it cheaper visually or you can do it cheaper phenotypically. Okay? Then you have to try to minimize the infrastructure, the investment in infrastructure, and try to use outsourcing if you can. That is a very good way of doing it in a viable way economically. And then you have to, if you're a breeder or if you're a pathologist and you just barely know enough about molecular marker to be dangerous, you have to enlist the services or the help or the collaboration of experts. I mean, it requires some expertise, and not everybody understanding in general, like I do, how marker work does not make you an expert, and you need to really have an expert to help you in this. Okay? And then, as I just said, if you want to implement molecular marker in your program at a regional or national level, make sure you don't take resources from your phenotypic selection, from your breeding program, conventional breeding program, to put into molecular marker. It has to be resources that are new, because you cannot strengthen one side by weakening the other. And that's a mistake that has been done in many national programs, including in developing countries, investing in millions of dollars in labs that ended up not producing anything, just because the breeding programs themselves were so weakened and became so unviable that they could not use these molecular tools anymore. So again, you need to have a strategy, you need to discuss it with your colleagues. It's a great tool. It can really multiply exponentially the efficiency of a breeding program, but it has to be done under certain conditions. With that, I thank you very much for your attention, and I will take any question whenever we can.